The challenge of developing an effective HIV vaccine has persisted for decades, driven by a complex interplay of scientific barriers and the urgent need for global solutions. Despite all the progress made in understanding the virus, finding a vaccine that works remains one of the most difficult tasks in modern medicine. Human immunodeficiency virus, or HIV, which is the causative agent of acquired immunodeficiency syndrome, also known as AIDS, continues to represent one of the most significant global health challenges of the modern era. Even though there have been remarkable advances in antiretroviral therapy that have transformed HIV infection from a fatal diagnosis to, a manageable chronic condition, the virus still exacts a devastating toll worldwide. According to recent data from UNAIDS, about 39 million people globally were living with HIV at the end of 2022, with around 1.3 million new infections occurring every year. The development of a safe, effective, and affordable preventive HIV vaccine remains a critical public health priority for several compelling reasons. First, while antiretroviral therapy has proven highly effective at controlling viral replication and preventing disease progression, it requires lifelong adherence and does not eliminate the virus from the body. Second, despite expanded access to pre-exposure prophylaxis and other prevention tools, new infections still occur at unacceptably high rates, especially in resource-limited settings and among marginalized populations. Third, the economic and social burdens of maintaining lifelong treatment for millions of people are substantial and honestly potentially unsustainable in the long term. A preventive vaccine really represents the most promising approach to controlling and potentially ending the HIV pandemic. However, despite over 4 decades of intensive research efforts and billions of dollars in investment, an effective HIV vaccine remains elusive. This article examines the complex biological challenges that have impeded HIV vaccine development, reviews current strategies and clinical trials, and highlights promising avenues for future research. HIV belongs to the lentivirus genus of the Retroviridae family. The virus primarily targets CD4 positive T lymphocytes, which are critical coordinators of adaptive immune responses. The infection process begins when the viral envelope glycoprotein known as GP120 binds to the CD4 receptor on the target cell surface. This interaction induces conformational changes in GP120 that enable subsequent binding to a co-receptor, typically either CCR5 or CXCR4 chemokine receptors. Following co-receptor binding, the viral transmembrane protein GP41 undergoes structural rearrangements that drive fusion of the viral and cellular membranes, allowing the viral capsid to enter the cytoplasm. Once inside the cell, the viral enzyme reverse transcriptase converts the HIV RNA genome into double-stranded DNA. This process is inherently error-prone, lacking proofreading capability and generating about 1 mutation per genome per replication cycle. The newly synthesized viral DNA, along with viral integrase and other proteins, forms the pre-integration complex, which is transported to the nucleus. Integrase then catalyzes the insertion of viral DNA into the host cell's chromosomal DNA, establishing a stable provirus that can either actively produce new virions or remain dormant, depending on the activation state of the infected cell. After integration, the HIV provirus utilizes the host cell's transcriptional machinery to produce viral RNAs, which serve both as genomic RNA for new virions and as mRNA for viral protein synthesis. Initial transcription yields predominantly multiply spliced mRNAs encoding regulatory proteins like TAT and reverence. TAT enhances transcriptional elongation from the viral promoter, while REV facilitates the nuclear export of unspliced and partially spliced viral RNAs encoding structural proteins. Assembly of new virions occurs at the plasma membrane, where viral GAG polyproteins orchestrate the incorporation of viral genomic RNA, enzymes, and envelope glycoproteins. Immature virus particles bud from the cell surface and subsequently undergo maturation through proteolytic processing of GAG and GAG-POL polyproteins by the viral protease, forming the characteristic conical capsid of infectious HIV virions. HIV has evolved multiple sophisticated mechanisms to evade host immune responses, which present significant challenges for vaccine development. Extraordinary genetic diversity. HIV-1 exhibits remarkable genetic variability with global circulation of multiple groups, M, N, O, and P, and within group M alone, at least 9 genetically distinct subtypes, including A through D, F through H, J, and K, as well as numerous circulating recombinant forms. Within a single infected individual, HIV exists as a swarm of genetically related but distinct variants, also known as quasi-species. This diversity is honestly primarily driven by the error-prone nature of reverse transcriptase, the high replication rate, producing about 10 billion virions daily in untreated infection, and recombination between co-infecting viral strains. Glycan shield. The HIV envelope glycoprotein is heavily glycosylated, with N-linked glycans making up approximately half of its molecular mass. These host-derived glycans form a dense shield that obscures conserved viral epitopes from antibody recognition, effectively camouflaging potential neutralization targets. Conformational masking. Critical conserved structures on the HIV envelope, such as the CD4 binding site, are recessed and sterically protected, becoming transiently exposed only during receptor binding. This conformational masking, limits the window of opportunity for antibody neutralization. Establishment of latent reservoirs. HIV establishes persistent infection in long-lived memory CD4 positive T cells and other cellular reservoirs. In these cells, the integrated provirus may remain transcriptionally silent, rendering it invisible to immune surveillance mechanisms that target actively infected cells. Rapid escape from immune pressure. The virus can, quickly develop mutations that allow escape from both antibody and T cell responses. When effective immune responses do develop, they typically target regions of the virus that can tolerate mutation without significant fitness costs. Downregulation of MHC class I, HIV NEF protein downregulates surface expression of MHC class 1 molecules on infected cells, thereby reducing recognition by cytotoxic T lymphocytes. Depletion of CD4 plus T cells. By specifically targeting CD4 positive T cells for infection, HIV progressively depletes these critical orchestrators of adaptive immunity, compromising the overall functionality of the immune system. The combined effect of these evasion mechanisms creates a formidable challenge for vaccine development, as an effective vaccine must overcome multiple layers of viral defense simultaneously. The development of an effective HIV vaccine faces several interconnected scientific challenges. Absence of documented natural immunity. Unlike many viral infections for which vaccines have been successfully developed, there are no documented cases of complete spontaneous clearance of HIV infection in humans. This absence of natural protective immunity unfortunately provides no blueprint for vaccine developers to mimic. Lack of correlates of protection. Researchers have yet to definitively identify which specific immune responses, like antibody levels, T-cell functions, and so on, reliably predict protection against HIV infection or disease progression. Without established correlates of protection, vaccine development proceeds without clear immunological endpoints. Viral genetic diversity. The extraordinary genetic diversity of HIV globally and even within individual hosts necessitates vaccines that can induce immune responses against multiple viral variants simultaneously. Immune evasion mechanisms. As detailed previously, HIV has evolved sophisticated strategies to evade both humoral and cellular immune responses, making it difficult for vaccine-induced immunity to effectively control the virus. Limited animal models. While non-human primate models using SIV or SHIV, that is simian human immunodeficiency virus, provide valuable insights, they incompletely recapitulate HIV pathogenesis in humans, complicating the translation of preclinical findings. Researchers have explored multiple vaccine platforms and strategies in the quest for an effective HIV vaccine. Live attenuated vaccines. This approach uses weakened forms of the virus that can replicate but not cause disease. While this strategy has been successful for many viral diseases, safety concerns regarding potential reversion to virulence or recombination with wild-type HIV have largely precluded its development for HIV in humans, despite demonstrating efficacy in non-human primate models. Inactivated vaccines. These vaccines contain chemically or physically inactivated virus particles. Early studies with inactivated SIV vaccines in non-human primates failed to induce protective immunity and in some cases even enhance infection, leading researchers to abandon this approach for HIV. Subunit and recombinant protein vaccines. These vaccines deliver specific viral proteins, typically envelope glycoproteins, to induce antibody responses. The RV144 trial in Thailand, which showed modest 31.2% efficacy, used this approach in combination with a viral vector prime. Viral vector-based vaccines. These platforms use modified, non-pathogenic viruses such as adenovirus or modified Vaccinia ankara to deliver HIV genes, primarily aiming to induce cellular immune responses. Recent major trials using this approach include HVTN702, HVTN705, also known as Imbokodo, and HVTN706, or Mosaico, which unfortunately did not demonstrate efficacy. DNA vaccines. These deliver plasmid DNA encoding HIV proteins directly into cells, where the DNA is transcribed and translated to produce viral proteins that can stimulate immune responses. While safe, DNA vaccines alone have typically shown limited immunogenicity in humans and are often used in prime boost regimens. mRNA vaccines. Building on recent successes with COVID-19 vaccines, mRNA platforms are being explored for HIV vaccine development, offering advantages in rapid production and potent immunogenicity. Mosaic and conserved element approaches. These strategies aim to address viral diversity by including either computationally optimized sequences that maximize coverage of global HIV variants, Mosaic, or by focusing immune responses on functionally constrained viral regions that cannot easily mutate, known as conserved elements. Over the past several decades, a number of large-scale HIV vaccine efficacy trials have, provided some really important insights, even though the primary outcomes were, often disappointing. The STEP and FAMBILI trials, running from 2007 to 2009, evaluated an adenovirus vector vaccine, specifically Ad5 expressing HIV internal proteins. Not only did this vaccine fail to protect against HIV acquisition, but interestingly, vaccinated men who already had immunity to Ad5 actually showed increased HIV acquisition. This outcome really highlights potential risks associated with vector-based approaches. Moving on, the RV144 Thai trial in 2009 used a canarypox vector prime, known as Alvacchivi, followed by a GP120 protein boost, or AIDS-VAX-BE. This trial demonstrated modest efficacy at 31.2%, providing the first evidence that vaccination could prevent HIV acquisition in humans. Further analysis suggested that non-neutralizing antibodies targeting the V1-V2 region of GP120, as well as ADCC, antibody-dependent cellular cytotoxicity responses may have contributed to protection. Then, the HVTN-702 trial, conducted from 2016 to 2020, attempted to build on the RV144 results by testing a similar regimen, but adapted to HIV subtype C, which is predominant in Southern Africa. This trial was stopped early for non-efficacy, showing that strategies successful in one population and against one viral subtype may not necessarily translate to others. The HVTN-705, also called Imbokodo, from 2017 to 2021, and HVTN-706, or Mosaico, from 2019 to 2023, both evaluated Mosaic immunogen vaccines delivered via adenovirus vector prime and protein boost. Despite promising preclinical results in non-human primates, both trials were discontinued due to lack of efficacy. Altogether, these trial outcomes really underscore the formidable challenges facing HIV vaccine development and, honestly, the limitations of current animal models in predicting human efficacy. Despite these setbacks, several promising approaches have emerged that might, hopefully, overcome some of the historical obstacles to HIV vaccine development. Broadly neutralizing antibodies, or BNAbs, are a key area of focus. Unlike the strain-specific neutralizing antibodies that are typically elicited by viral infections, broadly neutralizing antibodies can neutralize a wide range of HIV strains across multiple subtypes. These antibodies target conserved regions of the envelope glycoprotein, regions that are, really critical for viral function and therefore less tolerant of mutation. Key BNAb targets include the CD4 binding site, the membrane proximal external region, or MPER, of GP41, V1, V2 glycan-dependent epitopes, V3 glycan-dependent epitopes and the GP120, GP41 interface. While BNAbs develop naturally in about 20 to 30% of HIV-infected individuals, they usually emerge only after several years of infection, following extensive coevolution between the virus and B-cell responses. Recent advances in structure-based immunogen design are aiming to guide B-cell development toward BNAb production much more efficiently than what happens during natural infection. Now, speaking of novel immunogen design strategies, high-resolution structural information about the HIV envelope has enabled rational design of stabilized native-like trimers, such as SOCIP constructs, that present neutralizing epitopes more effectively while occluding non-neutralizing ones. Germline targeting approaches are another exciting area. These strategies aim to activate B-cell precursors that have the potential to develop into BNAb-producing cells, using sequential immunization with engineered immunogens, including germline targeting, intermediate and booster immunogens. Epitope-based vaccines offer a different angle. Rather than using the entire envelope protein, some approaches focus on presenting specific conserved epitopes in ways that really boost their immunogenicity. On the technology side, mRNA platforms have come into play. Building on the success of mRNA vaccines for COVID-19, researchers are now exploring mRNA platforms for HIV vaccination. These approaches offer several advantages, such as rapid production and iteration of candidate immunogens, strong induction of both antibody and T-cell responses, the ability to deliver multiple immunogens at once, and the potential for repeated boosting without anti-vector immunity. Recent preclinical studies have shown that mRNA-delivered HIV envelope trimers can induce potent neutralizing antibody responses in animal models, which suggests real promise for human applications. Beyond that, there are alternative and complementary approaches as well. Passive immunization, which involves direct administration of broadly neutralizing antibodies, has shown promise for both prevention and treatment in clinical trials. Long-acting formulations and combinations of BNAbs could offer practical alternatives while vaccine development continues. T-cell vaccines are another avenue being explored. While neutralizing antibodies remain the primary focus for preventing acquisition, vaccines designed to elicit potent and broad T-cell responses might help control viral replication and prevent disease progression, even if infection does occur. Finally, combination prevention, integrating partially effective vaccines with other prevention modalities like PrEP, could achieve population-level impact through complementary mechanisms. After more than 4 decades of research, the development of an effective HIV vaccine remains, without a doubt, one of the greatest challenges in medical science. The unique biological properties of HIV, its genetic diversity, sophisticated immune evasion mechanisms, and its ability to establish persistent infection have really frustrated conventional vaccine approaches that have worked against other viral pathogens. Still, significant progress has been made in understanding the virus's vulnerabilities and the immune responses needed for protection. The discovery of broadly neutralizing antibodies has shown that in principle, the human immune system is capable of producing antibodies that can neutralize diverse HIV strains. Advanced structural biology techniques have illuminated the atomic level details of the HIV envelope, enabling rational immunogen design. New vaccine platforms, especially mRNA technology, offer, unprecedented flexibility in both vaccine design and production. Looking ahead, the path forward will probably require innovative approaches that induce immune responses that are qualitatively different from those generated by natural infection. Sequential immunization strategies, which aim to guide B-cell development toward broadly neutralizing antibody production, represent a really promising direction. Combination approaches that engage multiple arms of the immune system at the same time may help overcome the virus's redundant evasion strategies. While the timeline for developing a highly effective preventive HIV vaccine remains uncertain, the scientific foundation for success is honestly getting stronger all the time. In the meantime, integrating partial vaccine efficacy with existing prevention tools could help reduce HIV incidence, while research toward more effective vaccines continues. The lessons learned from HIV vaccine research have already transformed our understanding of viral immunology and vaccine development, with implications that really extend far beyond HIV itself. References for the information presented in this video are available for further reading.